Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. I'm your host Talia. Welcome to the BS level biology program with course code 4401. Students, I hope this series is broadening your vision and giving you new concepts to ponder upon. I'm sure by now you are ready for today's lecture. So now let's welcome our expert, Dr. Muhammad Azad. Assalamu alaikum, sir.
Wa alaikum assalam.
So what's the topic for today's discussion?
Talia, today we are going to continue our previous topic, a very important aspect of our previous topic, that is the control of gene expression. Students, in our last lecture we have gone with how the proteins are being synthesized, and today we are going to look at its control mechanism. So what is the control mechanism? All of you already know that there are trillions of cells. All of these cells, they are different in structure and they are performing different function. We discussed this idea before also, that how they are different. All of these, they are going to originate from a single cell, that is the zygote. By the process of mitosis, and the beauty of mitosis that they are going to produce identical daughter cells. Then all our body cell must be identical. But a cell of our muscle is different than a cell of RBC. RBC is different from a cell in our eyes, that is rods and cones of retina of eye, and that is different from a cell lining our intestine or the beta cell in the islets of Langerhans or pancreas. All of these cells, they are different in their structure and they are performing different functions. How it is possible? All of these are having exactly the same number of genes, same number of chromosomes, everything is same. Then how can they be different? God has given the mechanism of switching on of the certain gene and switching off the certain gene. Let's say in case of beta cell of islets of Langerhans of pancreas, the genes for synthesis of insulin is switched on, and that is signed a function of synthesis of insulin. So that start producing insulin. The gene for pepsin is also present in that cell, but that is switched off. While in case of a cell lining the gastric mucosa, so there is a gene that is concerned with the synthesis of pepsin is switched on. Insulin gene is also present in the stomach, but here it is switched off. So today we are going to look in details how the genes are switched on and off to have a specific action at a specific moment. So going with our slideshow, this is the continuity of our previous programs, that is genetics, linkage and crossing overs, and today's topic belongs to the molecular genetics, that is the regulation of gene expression. So this is the DNA which is going to store all the information in the form of genes, and then these genes, they are being expressed, switched on to make specific protein, and then these proteins are going to give us a specific trait. Let's say in case of hand, a specific feature of hand that is given by this gene. So the code is there within the cell, and within the cell in the nucleus, and this code is being expressed in the form of genes. So gene expression is going to give this color of eye, the blue color of eye. So traits are controlled by expression of the genes. Each cells in humans contain all the genetic material for the growth and development of a human. Some of these genes will be needed to be expressed all the time. So they are involved in the vital biochemical processes such as respiration, which is occurring all the time, and other genes, they are not expressed all the times. So if they are required, they are switched on, and if they are not required, they are switched off. So they are regulated according to the need of the body. Starting with the processing of gene information in prokaryote versus eukaryotes. So basically gene information is processed differently in prokaryotes and eukaryotes, because the prokaryotes, they are going to lack the nuclear envelope. Messenger RNA can associate with ribosome in the cytoplasm as the messenger RNA is being produced. So this is the messenger RNA. At the same time translation is occurring, these are the ribosomes, and these are 70S ribosome in case of prokaryote, 3 polypeptide chain, 1, 2 and 3 they are being produced, and these are produced by the 3 genes. And in case of prokaryote, a group of genes which are going to be expressed at the same time by the same regulator, that is called as operon, and this operon is basically the unit in case of prokaryote. In case of eukaryotes, because the membrane bound nucleus is there, so here first the messenger RNA, the full copy is being produced, and then the processing of messenger RNA is done. The inactive parts, the introns are being cut down, the exons, the active parts, they are being joined together, and then 5' cap and 3' polytail, that polyA tail that is given, and then this is going to pass through the nuclear pore outside and associate with the large and small subunit of ribosome, giving us a polypeptide chain. In case of eukaryotic cell, so the process of transcription occur in the nucleus and translation occur outside the nucleus in the cytoplasm. Going with the regulation of protein synthesis and metabolism, genes are regulated to be active only when their products are required. This is very important. So as per requirement, gene expression is there. In prokaryotic cell, this regulation is coordinated by operons, and operons are defined as a set of genes, all of which are regulated as a single unit. Operons, they were discovered by Jacob, Monod and Loff, and they are offered Nobel Prize on the discovery of these operons. And operon is a group of the genes that are transcribed at the same time. They usually control an important biological process. Going with the Lac Operon or lactose operon, the Lac Operon consists of 3 genes, each involved in the processing the sugar lactose. One of them is the gene for the enzyme beta galactosidase. This enzyme hydrolyzes lactose into glucose and galactose. E. coli bacteria can use either glucose, which is a monosaccharide, or lactose, which is a disaccharide. However, lactose need to be hydrolyzed and digested first, so the bacterium prefers to use glucose when it can be there available in the environment. So 4 possibilities are there. When glucose is present and lactose is absent, the E. coli does not produce beta galactosidase. When glucose is present and lactose is present, both are present. So E. coli prefer glucose and does not produce beta galactosidase. When glucose is absent and lactose is also absent, so both the products are absent, still there is beta glycosidase is not produced. But when the glucose is absent and only lactose is present, then the E. coli does produce beta galactosidase. Operons are classified into 2 varieties, inducible and repressible. Inducible operon is turned on by the substrate. A catabolic operons enzyme needed to metabolize the nutrients are produced when needed. The repressible genes in a series are turned off by the product. Synthesized anabolic operon, enzyme used to synthesize an amino acid, stops being produced when they are not needed. Dear students, so up till now we have gone with the inducible and repressible operons. What these are? Actually, in all the chemical reaction, there are certain substrates which are converted into products with the help of specific enzymes. So some of these operons, they are being regulated by the substrate. If substrate is more, then there is that the more of this, you can say the reaction should be carried out. Enhancement of speed should be there. So these substrate induced operon are called as inducible. They are going to increase the rate of reaction. If the product is more than our requirement, so this extra product itself is going to cause switching of this mechanism. So this extra product is going to switch off, or that is going to inhibit the rate of reaction. So these operons, they are called as repressible operons. Going with the details of both of these variety of operons in the form of slides and animations. So in this picture, we are going to look at these 2 operons. One is this Lac Operon, that is the lactose operons. So once you can say it is there in the active repressive form is going to block this operator gene once the lactose is inactive and it is being expressed, while in case of tryptophan is one of the product. So normally the inactive form of repressor is this molecule. Once the tryptophan is going to bind within this place, it is going to cause the active repressor, and this gene is being you can say the repressed. So this is the one of the inducible operon, and this is one of the repressible. This is one of the inducible operon, and this is one of the repressible operons. So going with the details of lactose operon or the Lac Operons, that is going to have three segments. The first segment is regulator. What the regulator is? It is the gene that codes for repressor. Then there is a central locus which is composed of promoters and operators, and then there are structural genes which are made up of three genes in this case. One of the gene is beta galactosidase, that is going to hydrolyze lactose. Second, it is the permease which brings lactose across the cell membrane, and the third enzyme that galactosidase transacetylase, the function of this enzyme, they are uncertain till this moment. So normally the Lac Operon, that is switched off. In the absence of lactose, the repressor binds with the operator locus and blocks transcription of downstream structural genes. Lactose turns the operon on. Binding of lactose to the repressor protein changes its shape and causes it to fall off the operator. RNA polymerase can bind to the promoter. Structural genes, they are being transcribed. So here let us say this is the situation. This is the regulatory gene that is going to produce this repressor protein, and this repressor protein is going to bind here at the operator to block the path. This RNA polymerase cannot go beyond that, and there is no RNA, and there is gene is being switched off. When the lactose is present, lactose combined with this repressor, and this shape, this is being changed. Now this shape is not going to fit here at the operator genes. So once it is not going to fit, it is going to come out, and RNA polymerase is going to transcribe all of these three genes Z, Y and A, and these are going to produce these three enzymes. So this is the animation about the combination of switches in the Lac Operon. In E. coli bacteria, two regulatory sites are there, the cap binding sites and the operator functional sites, and both of these, they are going to function together to ensure the three proteins required for the growth on the bacteria. If the lactose is present,